We change a little bit scenario now. We are targeting the apoptotic pathway, always in CLL. And that's the title of the session, with only 2 talks, but great talks, one by me, one by Steve. And then, so the first one is overcoming the challenges in targeting apoptotic pathway. So I will leave to Steve the clinical data. I will try to give some rationale, some background, and again, I will try to foster some discussion. My baby is no good because we are wasting time. So overcoming, so the idea of BCL2 apoptosis targeting apoptotic pathway, of course, comes very easily when you think about follicular lymphoma, BCL2 overexpression due to the translocation. The translocation of T1418 is also present in some diffuse large B-cell lymphomas originating, transforming from follicular or de novo. And then we can have an amplification in the BCL2 locus, also in other diffuse large B-cell lymphoma, the ABC type, and also in some mantle cell lymphoma, if you believe it. So the idea to target BCL2, there has been a lot of approaches and attempts in the past with antisense and so on. The best way to go now is by BH3 mimetics, so molecules that bind to BCL2 and then disrupt the association with the pro-apoptotic molecules that then are free to act inside the cells and kill the cell. So here it is. So there has been a number of generation of products of BH3 mimetics. We skipped the first generation, which was IV, was a broad inhibitor IV. Now we move to direct to the second generation, where we have an orally available drug, which is navitoclax ABT263, and that was still a very broad inhibitors inhibiting BCL2, BCLX long, and BCLW. And the main problem with this was indeed that all the studies that have been performed show the grade 4 thrombocytopenia, which was inevitable, I would say, because it's on target effect due to the fact that the thrombocytes are heavily are kept alive due to the overexpression, to the expression of BCLX long. So the fact that it was targeted by this drug, then it led to really apoptosis of platelets. So so maybe counterintuitively, because we have been always educated in oncology that we should always try to hit more targets at a time, to hit more pathway in order to get more efficacy. Here instead, the company went backward, and so they made it narrower. They made the efficacy narrower. So they focused on BCL2 alone, creating the BT199 that is indeed not interfering anymore with the BCLX long, and also with any other member of the BCL2 family. And therefore, there was no more dose-limiting thrombocytopenia, at least in theory, but also then we will see in real life. And while instead they thought they could have still efficacy in leukemia and lymphoma. So the problem is, we didn't move the, okay. The problem indeed was that also navitoclax did not show really great results in follicular lymphoma, which was kind of, as Wyndham showed, was kind of surprising again, because based on the literature, based on our scientific knowledge, we would have bet on this. And instead, this was not true. And indeed, you don't see anything impressive. And also with the ABT199, the situation was not much different. A number of non-Hodgkin lymphomas have been treated, mantle cell, marginal zone, diffuse, follicular, Waldenstrom, and the follicular are the one in purple. And again, the response below or above 50% partial responses were only 4 out of 13, which is something that you definitely would not expect if in a disease where BCL2 is, we know, the central gene in the disease. That's probably because it's not indeed the central gene, because BCL2 is indeed translocated. So, but that translocation occurs in the bone marrow during the B cell development, so at the precursor B cells. So it's something that the cell acquire, one cell, that provides probably a survival advantage to that particular cell, which is a precursor B cell. Then it moves, becomes mature B cells, it goes into the germinal center during immune reaction, and there something else is happening. And then that particular cell is expanded and becomes a lymphoma. So, but as shown also, I think as a proof of the fact that these anti-apoptotic inhibitors are not so effective, are really suggesting that BCL2 is not playing such a key role in when the disease is progressing. And indeed, we showed several years ago, almost 20 years ago, I would say this was my first paper published as a postdoc. So the cells, when follicular lymphoma cells could be rescued by death in vitro by stimulating with CD40 ligand. Stimulation, the CD40 ligand stimulation actually maintained high level of MCL1 or BCLX long. And as you can see here, the MCL1 and also the BCLX long was upregulated, really suggesting that the BCLX long and probably other molecules at that time we didn't know play also a relevant role to maintain the cell. And again, it's possible that microenvironment can have a role also in this lymphoma. So, but then we also learned, and this is definitely much younger as a study, that we already heard in previous talks that there are other gene aberrations in follicular lymphoma that likely accumulate with time. These are mainly in the methylation and control of the gene transcription, so the epigenetic mechanism. And indeed, so the idea probably with for treating follicular lymphoma, so to overcome the challenges, is that definitely the BCL2 inhibitors can have a role because the BCL2 molecule is there, overexpressed, but maybe one should think targeting the other gene mutations that are aberration that are occurring in follicular lymphoma, including the epigenetic machinery. It is also true that these HDAC inhibitors and any other molecules that are now available for clinical trials showed efficacy, but also a great level of toxicity. So that's the problem that should be overcome. But it could be really a target that can be worth targeting in follicular lymphoma. But instead, in contrast to probably what people thought before, when ABT199, so the BCL2 inhibitors, have been used in chronic lymphocytic leukemia, then we all saw this wonderful waterfall plots where, again, already spoiled by ibrutinib, idelalisib, virtually all patients responded, 90%, over 90% of patients responded. And why is that? Again, if we go back to the biology of the disease, we know that BCL2 is indeed overexpressed in all CLL, this bone marrow. And again, it is expressed not because of a genetic lesion, but because it can be modulated by CD40 ligand, by toll-like receptor ligation, by the stroma interaction, and so on. So again, it's something that is maintained by the microenvironment. It has also been suggested by Carlo Croce that the patients who are carrying the deletion 13q, they are missing MIR15 and 16, which are inhibitory of BCL2 transcription. So in the absence of MIR15, 16, then you have an upregulation of BCL2. So there could be also a genetic mechanism that can be acting maybe in 50% of the patient, but still in all CLL patients there is the BCL2 overexpression that can also be driven by the microenvironment. Regardless the mechanism, BCL2 is relevant in CLL and is acting in this very moment, I would say. So whenever the disease is occurring, the BCL2 is acting and is present at high level. So this led to these very nice responses. You will see much more in detail by Steve, but just to see that 80% of the patients I showed you already before responded with 1/4 of the patient achieving complete remission and including MRD negativity. But then we move forward because again, we don't know if we are curing the patient with this drug. Steve will tell us. But again, probably the BCL2 story is not the end of it. We have also other genes and other protein, anti-apoptotic proteins that are also acting and working CLL due to the microenvironment. So there is MCL1. MCL1 is upregulated by CD40 ligand, is upregulated by the stroma, as I said before, and it provides chemoresistance also. So again, we don't have to focus our attention only on one molecule, and maybe the contemporary targeting of both BCL2 and MCL1 could be a strategy for the future. How can we do that? Because it is true that ABT199 has been used only a couple of years, or 2 or 3 years, but there are already cases that can be resistant to ABT199, and especially in the lab, cell lines have been created that are resistant to ABT199, and what they show is indeed that there is an increase in MCL1. So it is somehow the inhibition of BCL2 is counteracted by the increase of MCL1 expression. So of course one can target directly MCL1 and downregulate also the MCL1, but another strategy would be to use here GS1101, which is nothing else than idelalisib, because there is an activation of MCL1 overexpression is due to an hyperactivation of the PI3 kinase AKT pathways that is compensatory compared to the downregulation of BCL2. And so by hitting this pathway, then you would decrease also MCL1. So again, how these new drugs can be combined with a value for the patient. Just because we have to mention ibrutinib, the same thing happens with ibrutinib. Indeed, so whenever you hit, if you have a cell line that is ABT199 resistant, then you have a high level of AKT, and then if you combine with ibrutinib, then you have a downmodulation of AKT, and at the same time also downmodulation of MCL1. So just to make you understand that we have always to look at different molecules. We cannot think to cure the disease probably with one molecule only, so we should find a way to target more molecules, and probably by combining molecules, drugs that we already have available, we could have a beneficial effect. Thank you for your attention.